for the day, Dr. George Church, to please officially release the BioRiddle video on the website and therefore inaugurate BioRiddle. So you will find the link in the chat box. You just have to release it by pressing the play button on the official website.
I pressed the button.
Yes.
Yes, I've done that. Yes. Do you
Thank
want
you
to
so
share
much,
it as
sir.
well?
Yes. It is so BioRiddle is officially inaugurated. Moving on, it is indeed a proud moment for me to be introducing our keynote speaker for the day, Dr. George M. Church. For a student of science myself who loves molecular biology and genetics, so this is a fangirl moment for me. George M. Church, sir, PhD, is a professor of genetics at the Harvard Medical School, a founder member of the Rice Institute, and director of personalgenomes.org, the world's only open access information on human genomic, environmental, and trade data. Dr. Church is known for pioneering the fields of personal genomics and synthetic biology. He developed the earliest methods for the first genome sequence and dramatic cost reduction since then, contributing to nearly all of the next-gen sequencing methods and companies that we know of. His team invented CRISPR for human stem cell genome editing and other synthetic biology techniques and applications, including new ways to create organs for transplantation, gene therapies for age reversal, and gene drives to eliminate Lyme disease and malaria. Sir is director at IRPA and NIH Brain Projects at the National Institutes of Health Center for Excellence in Genomic Science. He has co-authored more than 515 papers and has 130 patent publications. Also a book, Regenesis. The honors include the Franklin Bower Laureate for Achievement in Science, Times 100, and election to the National Academies of Science and Engineering. So it would not be fair on my part to stand any more between you and the audience. So I hand it over to you, sir, for your keynote address.
Thank you very much for such a kind introduction, and it's wonderful to be with so many of you today. I'm just going to share my screen. And this is about Darwin. This is about accelerated evolution and how we achieve it with multiplexing library and artificial intelligence. I have a conflict of interest webpage if you're interested in that. And certainly it's a celebration of all the things represented here today. Most importantly is open access and equitable distribution of new technology. That requires getting as close to $0 as possible for each of these technologies. Key examples are reading and writing of DNA. Here, these two lines have shown that we've gone about 60 million fold in just a few years. We've gone from a $3 billion human genome reading, for example, which was not a very good genome because it was only half a genome. And now it's 300, soon $100 per genome, and we hope to keep driving that price down until everybody can afford it if they want it. Now, the way that we've done this is not via automation. That you would have there's quite a bit of room for improvement. Not via automation or parallelism. It's not filling a room full of identical devices that each cost the same amount, but by reducing that cost millions fold by what we call molecular multiplexing. It's analogous to telecommunications multiplexing, but in this case you have hundreds to trillions of reactions in a single droplet being treated through a series of of chemical or biochemical reactions with the same efficiency and cost as doing one reaction. So a trillion reactions for the price of one. And this can be not only used for reading and writing DNA, but for editing, developmental biology, and so forth, as you'll see in a moment. Again, not manual. Manual and automation is basically the same number of tips, the same amount of solution. You have to completely rethink it via multiplexing. So my first example of multiplexing is in protein engineering. And we've seen a revolution in protein engineering, not just because we can multiplex, but because we also can use machine learning and synthesis of libraries. So you really need to have all three to make the best drugs, the best enzymes, and so forth. And for drug development, for example, we can multiplex in at the expensive end of the clinical trials, as close to humans and including humans, where the relevance is higher, the safety and efficacy is more relevant. Now this protein engineering is based on about 4 papers we published recently here, 2 of them, that showed how you could use deep learning, machine learning, to train on very large sets, essentially all known protein structures and sequences, to allow you to leverage small studies on small numbers of proteins, and then loop that up into millions of proteins. And so we can use this directed evolution where we don't synthesize random libraries, but very intelligently designed libraries, using knowledge from tens of millions of natural proteins. And this is work spearheaded by Ethan Alley and Serge Bijlois. This can be then used, this machine learning protein design can be used for testing billions of variants to get new enzymes, and this is what Nabla Bio is doing. Serge and Francis are doing this. It can also be used for marking proteins, so you can mix large proteins in an animal, and via their barcodes on the proteins, you can figure out which of them are being safe and effective or going to a particular place in the organism. And I'll show you an example of that in just a moment. Pierce, Gleb, and Shane spearheaded this in a company called Manifold Bio. Now, the third example of protein engineering is actually we're starting to get it a little bit bigger. We're going to work our way up from protein engineering all the way up to ecosystems. But this one is viruses. So it's a virus capsid protein, which we use to deliver gene therapies. AAV is by far the most approved gene therapy vector, but it has room for improvement, considerable room for improvement. And what we did was we made over 1 million different capsid designs based on first going through and systematically changing every amino acid in the protein to every other possible amino acid. This is grist for the mill. This is a primer for the pump of machine learning. You need to have lots of examples of, and we can classify each of those changes as to whether they're more or less active in a variety of different tissues. So we can inject a library into an animal and harvest different tissues and determine exactly what cells it's going to and how efficiently it's doing that. All of them 1 million at once. Pierce and Eric spearheaded this and launched Dino Therapeutics to do it. This loop of synthesis, selection, Darwinian selection, DNA sequencing, machine learning, and so forth. The machine learning is decidedly non-Darwinian. And the synthesis of libraries. So I'll show you some data on how we can use these libraries for getting at tissue specificity. Here, the tissue specificity of tropism can be measured by harvesting and then measuring out the barcodes for each of the different tissues, the barcodes of the viruses. These are essentially gene delivery. These are not real viruses, they're just capsids. And you can see this heat map where red means very strong and blue means weak relative to the wild type gene delivery vectors, and they differ from tissue to tissue. Here's 6 tissues, we've done more. The x-axis is the position along the viral genome, and the y-axis is every possible amino acid substitution. This is published in Science. and is a paradigm for how we can study a lot of different tissue tropisms, immune reactions. We want the vectors to avoid the immune system um, in therapeutic settings. So that's how we can make viruses better for gene therapy. How can we make ourselves resistant to viruses? And the range of avenues that we can take go from virus knocking out one receptor at a time. The problem is some viruses have multiple receptors. Many of those receptors are essential for the life of the cell. So you can't just knock out viral receptors. We can also, in some cases, the viruses are endogenous, and we can knock those out, and I'll show you an example in a moment. The most profound, and I think general, we think we can make any organism resistant to all viruses, as we have done for one industrial microorganism, E. coli, changing all of the UAGs throughout the entire genome, 321 edits simultaneously to achieve that. We do editing by a variety of methods. You can consider genome modifications broadly to be addition of genes, which is classic gene therapy, subtraction, which is CRISPR and other nucleases, precise editing, which are, I would say, deaminases and homologous recombination, and epigenetics. And I'll show you quickly examples of each of these. Together with Jennifer Doudna and others, we've started some companies to bring these to everyday citizens. Now, for precise editing, this cannot be easily done by just double-strand breaks. It requires something a little more finesse, especially if we're making a lot of them at once. If you do a lot at once, double-strand breaks will scramble the genome. It will cause toxicity. The cells will often apoptose and die. Here are two examples of precise editors. The lambda red recombinase, which is working very well in a variety of organisms, finally, after 2 decades of it only working in one organism, it now works in many. And these serine and tyrosine integrases, which have always worked in many organisms. And this is work from Kundar, Labar, and Weinberg. Probably the best example of applying both of those, both the recombinases and the integrases in the previous slide, into one coherent project where the design was effectively to completely change the codon usage in the whole organism, so it changing one of the 64 triplet codons. Not just because we can, but because it has 3 practical applications. We can do new amino acid chemistry, we can do biocontainment, and most importantly, this multivirus resistance. We think we can make any organism resistant to all viruses. In this case, we picked a particularly wimpy codon that wasn't intended to provide multivirus resistance, but it turns out that it did. But by going to a transfer RNA codon rather than a stop codon, we can do even better. And these are Science and Nature papers from LaJoy, Mendel, and Ostrov. Ostrov is still leading the project. So we did 321 codons, completely finished that project, shows virus resistant. I'll show you that data in the next slide. And now we're working on 62,000 codons, which will essentially 7 of the 64. Here are the first one in red, the 7 in red and blue, and then an effort from another lab involving another 2 in green. This is a codon table where the numbers are the number of instances of that codon for that amino acid throughout the genome, 64 different codons. The smallest number you can see is 321, and that's the one that we changed. And here's blowing up that section. Again, the 321 we reduced to 0, here in white 0, by moving it over into other stop codons, which are synonymous. And then tested them with multiple viruses. This was done in our lab and also in Farron Isaac's lab, which is in this Cell Systems paper, where you can see all these zeros are cases where we're starting with high titer virus, up to 10 to the 12th plaque-forming units per mil. It drops down to 0. We recover no viruses for all these zeros, very significant. And we think that by this sort of method, we should be able to make multiple organisms resistant to all viruses, not just a few, and so resistant that they can't revert because it would require hundreds of coordinated changes in a highly non-Darwinian fashion. This is non-Darwinian because we did synthesis and recombination. Now that involved 321 edits throughout the genome. I'm going to switch now to humans, where we start with a smaller number of edits and then work our way up to a larger number. We're going to start with like 1 or 2 edits and make it up to 22,000. So here is one of the first in the clinic for editing was adding to the chimeric antigen receptor in T cells, which attack B lymphomas and leukemias based on their surface antigens. You need to edit them a little to make them more effective, to avoid the host immune system, to avoid some of the chemotherapies that might accompany it. And this can be anywhere from 1 to 5 different edits listed here, these different genes, and they can be edited by any of the 3 nucleases, and this is the first child, I think, to benefit from this CAR T therapy using TALENs to do the editing, but it could be using fingers and CRISPR as well. Now that's on the order of 2 edits, plus the CAR T itself, which is not really an edit, it's an addition. Let's move from 2 to 42. Now for 42, this is also of clinical significance. Here, the goal is organ transplant, and in addition to there being a worldwide shortage of organs, especially high-quality ones, there's a need for enhancing the organs so that they don't succumb to the same disease that the first one did. If it's you want to make the organs as good as they can be in terms of resistance to metabolic disorders, resistance to pathogens, to senescence, and so forth. And so it's a huge opportunity to make organs that are better than you could get from a human being in order that the organ will last. And there was a wish list that accumulated over 20 years. We were not the pioneers in this. We came in to help out, and I think we've addressed all the genes on everybody's wish list at this point, 42 of them, all in 1 strain of pigs. We did it in cells and then transferred the nuclei into embryos of the pigs, and you can see here hiding in the upper right corner one of those. We now have 2,000 of these pigs that have been heavily edited, hitting the sugars, clotting factors, major histocompatibility, other immune functions, complement. And we even knocked out all of the endogenous retroviruses, because this is one of the concerns of the FDA, is that every cell in every pig in the whole world makes viruses that will infect human cells and then spread to other human cells. So an immune suppressed transplant recipient, this is particularly troublesome. And so we eliminated all of them, and so hopefully that will not be a problem. And we've shown that they do not get reinfected. So we eliminate them by just mutating the polymerase gene. We left the envelope gene intact, which is helpful for providing resistance to reinfection. Jim Markman at the Massachusetts General Hospital is testing these in primate trials, and it looks very good, on the order of 300-day survival. Luhan Yang spearheaded this as a graduate student, a postdoc, and now CEO of Kihan Biotech, and also co-founder of eGenesis. She's a brilliant person who helped also invent CRISPR. In order to pull this off, we had to, it's too many edits. Like I said, CRISPR is toxic, also other nucleases, and we had to coax the cells into surviving until we could get clones, and the clones we could transfer the nuclei. So even though they required all of this to keep them alive, they are healthy, and the pigs resulting from them are healthy and breeding. But a better way of getting the higher edits, so now we're going from 2 to 42 to more, is to not make double-strand breaks, or even NICs, is using deaminases, which Luhan Yang also pioneered, but mostly from David Liu's lab. We prefer the A to G editor. There's also C to T. So these are a very limited number of edits, and they tend to make a cluster of edits. But the A to G is the less toxic, and we eliminate the nicking enzyme. And we've applied this to an even bigger family of genes, not just the endogenous retroviruses, which, as I said, had about 25 genes in pigs, to these long interspersed repetitive elements or lines, where we have more than 20,000 of them in the human genome. And many of these repeats are involved in senescence, neurogenesis, cancer, and inflammation. They're not just passive junk DNA. They are actively involved, and a lot can go wrong with them. So the design of this experiment was to target a particular conserved region in the line elements, either with C to T transitions. So you can see here 4 red T's resulting from deamination of 3 red C's, 3 red C's to 3 black C's to 3 red T's, or A to G, a single A to G. It happens to be isolated, otherwise it would be a cluster just like with the C's. The A's are better mainly because they don't introduce nicks. The C turns into a U, which is subject to repair process, and it's more toxic. Also, the C to T enzyme, the deaminase, has more off-target effects. So off-target and toxicity due to nicking. So if we're aiming for a large number of edits, we need to get rid of every source of nicking, including the mismatch repair. And this is work from Corey, Oscar, Khalid, and Verena. So here is data from our record in pluripotent human stem cells. This is the hardest these are the hardest cells to work with. If you look at most of the CRISPR literature, even going back to our first paper in January of 2013, there's a tendency to use human cancer cells because they're easier, but they don't represent a therapeutic case, and so we use normal human stem cells. Here we achieved over 22,000 edits in a single cell, and these cells are stable to cloning and healthy. And we've characterized them by a variety of sequencing and other methods. And this is Raphael, Corey, and Oscar that did this work. That is our record so far. Is by no means an upper limit. I don't know what the upper limit is, but it bodes well for things like genome-wide recoding, which could potentially be done even in a therapeutic setting. Now to go to even higher numbers with different targets, so those are a repetitive family where they all have the same target. But if we want to have a more customized and more diverse set of targets, we need to have more more designer enzymes. They can either be guide RNAs in the case of CRISPR, or they can be new proteins in the case of other editing proteins. And in this case, we've put 50 guide RNAs into a single BAC, or bacterial artificial chromosome. The BAC can probably handle 1,000, but so far we've put 50 in, and also the cell can handle more than one BAC. So there's a lot of upside for the number of different guide RNAs we can put in now, and we've also seen that there's we can get up to 20,000 edits. So the challenge is to getting in that large number and having them all work, having an adequate amount of the editing proteins. As the number of guide RNAs goes up, it stays fairly flat in terms of the efficiency per guide RNA, so that's very hopeful. It indicates that we may be able to get to thousands of guide RNAs in the future. Now, I mentioned that we want to have senescence-resistant organs for transplants, because some of the organs are coming from animals that die at a younger age than humans. We would also like to have senescent-resistant drugs that confer senescence resistance or reverse aging aging reversal is an easier, shorter term experiment than is longevity or prevention, even though we would like those as well. And so we focus on reversing all these different diseases of aging. Here's 5 of them: high fat, obesity, diabetes, osteoarthritis, cardiac damage, kidney disease. We've added mitral valve disease, and we've shown this in mice, and now clinical trials are ongoing in dogs. After dogs, we'll start human clinical trials very soon. We've used as little as 3 genes, we've as many as 45 genes in early experiments, and we're constantly refining the list and dosage, relative dosage of these genes to hit all these diseases which have almost nothing in common other than the fact that they are all age-related. In fact, most morbidity and mortality has an aging component. Falling down and not getting up is age-related. Getting COVID-19 has an age component that almost everything does. And Noah Davidson, as a postdoc on my lab and now as founder of Rejuvenate Bio, is trying to bring this through the clinical trials to the people. And the final use of genome manipulation is epigenomics, where we're not actually changing the DNA at all, we're changing the way the DNA is regulated. And here are 3 of the many ways to regulate the cell, or the nucleus, the genome, microRNAs, RNA splicing factors, transcription factors. After a good bit of reflection, we chose transcription factors, and experiments indicated that these could be extremely powerful, and we haven't been disappointed once yet. And we've done now 290 different recipes to make 290 different cell types. And the idea is that we take these 1,700 transcription factors in expression vectors, where you can easily pop them via gateway into AAV, lentivirus, or piggyback. We like the piggyback the best because it has the biggest packaging size and the highest copy number, and hence the transcription factor that's expressed can overwhelm the epigenetic state of the cell at the time. So we've made, Alex and Peristu have set up GC Therapeutics to turn this into a cell therapy pipeline. It can also be used for testing non-cell therapies. As I said, we have 290 different cell type recipes so far. These are made by screening first for single transcription factors, but then for combinations that are hard to reach just from random combinations. So we use the transcriptome, ideally the single cell transcriptome of a target cell, to inform the kinds of combinatorial recipes we want to use, combinations of transcription factors in these expression vectors. Here are examples of 3 transcription factors with particularly simple recipes that make, respectively, endothelial cells, fibroblasts, and neurons. And you can see the very clean separation of these endothelial neurons and fibroblasts in this transcription factor heat map, where each of these is RNA along the y-axis and different transcription factors along the x-axis. And this just came out in Nature Biotech yesterday, so I urge you to take a look at it. Here's some of the cell types we make, and we can make these in combinations. These are this is kind of a snapshot, a cartoon of the inside of a brain. Key component of the brain is the blood vessels, the neurons, of course, but blood vessels, capillaries, astrocytes, microglia, which is part of the immune system, oligodendrocytes, which wrap the axon, as you would see in the white matter in your brain and the spinal column. All of these things need to work in fixed ratios and geometries. A lot of the geometry happens by self-assembly. The ratios we can engineer by ratios of different transcription factors in the precursor stem cells. Here's an example of 2 cells acting beautifully in concert to to, these are derived from the Personal Genome Project cell lines, as mentioned in the introduction to this talk. Personal Genome Project is an open access, open source project. Anyway, these stem cells were, here the axon of the neuron was wrapped around just the right number of times, typical of natural tissues, the white matter in the spinal column. So this myelin sheath is provided by oligodendrocytes, so that the neurons in the oligodendrocyte cells differentiate together, and we can also differentiate the endothelial cells to make capillaries, which are perfect 5-micron conduits for blood. We take all of those cells together in organoids and plant them into mice that have a genetic disease, is a demyelinating disease, is analogous to multiple sclerosis and other ones. And these mice can be rescued, here in the middle, relative to controls, by injection of these human organoids, which are endothelial cells, neurons, and oligodendrocytes. And again, this is Alex and Paris too. Now, as the last example I want to give here is what we call a flight recorder, a molecular flight recorder, where we're trying to record all kinds of activity going on throughout the body. And we can store on the order of the entire internet in the palm of your hand, in principle in DNA. In practice, we have successfully stored 4 trillion bits of information in just 30 nanograms of DNA scattered throughout a mouse body. And every single cell in the body, every nucleated cell has its own record of the developmental biology, and work from other labs since then have, using our system, have shown you can record hypoxia as well. So in principle, you can record any kind of physiological state like hypoxia or developmental lineage by using this recording device, which is basically, we used the CRISPR-Cas9 to do something unusual, which is have the molecular scissors act on itself and to continue to generate diversity. This is Reza Kolhar and Prashant Mali, these papers in Science and Nature Methods. I would urge you to take a look at it if you like the idea of recording throughout the lifetime of an organism. You can also get information on developmental biology, where we determine that the left-right decision making is after all the other decisions, like the forebrain, hindbrain decision during development. So I've mentioned at the very beginning that we're dedicated to bringing down costs radically, not just a factor of 2, but a factor of 60 million. This has been done for reading and writing DNA. What about therapies? Those tend to creep up in price rather than plummet exponentially. We think that there are 4 ways of doing this. For rare genetic diseases and infectious diseases, it can be preventative via sequencing, and either matchmaking or bioweather map. We should have at least the same level of interest in bioweather map as we have in regular weather maps, probably more. And it would have come in handy for this recent COVID problem. I mentioned machine learning already, and aging is a particularly large market, and hence, the larger the market, the smaller the cost, because you can spread out the R&D costs over many people. There's also gene drives. I'm not going to go into these. I urge you to take a look at it. This could apply to, mentioned in the introduction, could apply to malaria, Lyme disease, invasive species, locusts, so on. Has to be done extremely carefully, and almost all of our papers up to this point have emphasized ways that you can make the gene drive local. And finally, carbon sequestration and climate change. Almost all of carbon is in the Arctic. I will maintain both the ocean and the land masses, 1,400 gigatons on the land. And we can revive nearly extinct species, endangered species like the bison. Went from about 200 bison to 500,000 recently, and this was done mostly without government support, based on the economics of the species. We are working on reversing carbon into the soil and retaining the carbon that's already in the soil in the tundra, 1,400 gigatons. Put that in perspective. It's 9 or 10 gigatons per year. It's all of human activity put together. So the amount in the form of methane is enormous, and methane has a multiplier of 30 to 80-fold more damaging than carbon dioxide. So by returning the grass that was there 10,000 years ago, we can improve the reflected light, photosynthesis, and the packing of snow. And I just want to wrap up by thanking our lab members. I've been thanking them all as I go along on the slides, but also this is a lab meeting which is done virtually now, and many of the people are in laboratories where they have to be wearing masks even during lab meeting. And this is in support of and camaraderie with people around the world that have changed their life and in many cases suffered from this horrible disease. So thank you very much.
Thank you so much, George sir, for this amazing, amazing insight. Gene editing is the buzzword today, but you have taken us through the entire journey, shared your research with us. You touched upon everything from disease to environment and opened our eyes. I'm sure the young innovators uh, have spiked their curiosities about what research they could plan, what research they could join, and what is actually happening in the world out there. From anti-cancer to anti-disease, anti-aging properties, the list is endless. Thank you so much, sir, for sharing your research with us on this platform. I'm sure that the list of questions you can see and the comments in the chat will tell you very clearly how well the audience have taken this. I think we will have to compile the questions and email them to you, sir, for you to go through them later. Sir, I have a humble request with you. Could you please release the video when your screen is being shared so that,
When it's being
yes,
shared, yes,
yes, so
of
that
course.
we could have the official
I
release
thought
to
that
the
might be what you
audience.
wanted, yes.
Yes,
Okay.
yes. Thank you so much.
Uh, I'll do that right now.
Yeah,
Um,
the link is in the chat.
oh, I have it up. I had it
Yeah,
up before. I just
yeah.
I'm just trying to think of how to start it once I start sharing. There we go. Is that
Yes,
working?
sir. Yes,
Okay.
yes. Thank you so much.
It's lovely.
The official release of BioDiddle, everybody. It's wonderful to have BioDiddle being inaugurated at the hands of George Church, sir. I'd like the audience to give a big round of applause.
It's a wonderful space you've created. I think many amazing bits of science will come from here.
Yeah. A big round of applause for BioDiddle, its organizers, the team members, and everyone behind this effort. Audience, you could just share the applaud emoji. That would make us very happy. Thank you. Thank you so much.
That's a wonderful resources, wonderful facility. Thank you.
Thank you so much, sir. As we come to the close of this ceremony, I would like to give everyone an overview of what we have in store for you in